Now let us discuss about another topic in the bacterial genetics, that is transposition. So before starting towards the topic transposition, I want you to know the concept of transposable genetic elements. The transposable genetic elements is also called as jumping genes. So here what happened, there is certain structurally and genetically distinct segment of the DNA that have been identified and that have the ability to move around in the certain cut and paste manner between the chromosomal and the extra chromosomal DNA within the cells. So these DNA molecules are called transposons, also called jumping genes, um, and the mode of genetic transfer is called transposition. So what happens is that if we look at the plasmid, there is the certain portion of the plasmid that has the property of the transposon. That means this transposon can be copied towards the bacterial DNA. So what happens is that a jumping process involving a transposon. The transposon is the specialized segment of the DNA. So it can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA molecule. So that could be that DNA molecule could be the plasmid or chromosome. For example, let us say that the plasmid has the certain portion, this portion of the plasmid is transposition, you can see. So if we look at the bacterial DNA, there is certain target site. So this target site there I have denoted by the MT, and other bacterial DNA have the blackish color. So in this target site, this transposon get copied and pasted in this portion. So you can see is there the portion of this transposition is copied here. So in the target site there is the integration of the gene. So it is critical in creating the plasmid with multiple drug resistant and transfer across the species line. So it is very important phenomena. The transposition is the very important phenomena for creating the plasmid with multiple drug resistance. So due to this property, due to this transposition property, the bacteria, various bacteria and gets the property of the drug resistant and can acquire others various features that other bacteria has. So for example, let us take example of one bacteria. The plasmid of that bacteria has the property of the drug resistance, but if we look at the another bacteria, so if we look at the another bacteria, for example this bacteria doesn't have the property of the drug resistant. So after the combination of the bacteria, it means if the certain part of this plasmid get incorporated in this bacterial DNA, so the new DNA will be like this. So this certain part of the chromosome or the plasmid gets incorporated into the plasmid of the new bacteria, and the property of this drug resistance is acquired by this new plasmid. So this is the concept of the transposition. So basically, again I am going to say is that the transposition is the process involving a transposon. Transposon is the specialized segment of DNA which can copy and excise itself and then insert into the same DNA molecule or an unrelated DNA. It could be plasmid or the chromosome. So that I mean to say that this is very important for the acquiring various genetic materials from the different bacteria. I hope you understood this topic. Thank you.